Histology of salivary glands. The oral cavity has three major salivary glands, namely the parotid glands. These are paired glands and are seen anterior and inferior to the external ear. They are largest of all the three glands. Submandibular glands. These are also paired glands and are seen in the floor of the mouth inferior to the mandible. Sublingual glands. These glands are smallest of all the glands and are seen inferior to the tongue in the form of aggregate of small glands. A dense connective tissue capsule surrounds the salivary glands. This capsule sends septae into the glands, dividing them into numerous lobes and lobules. The salivary glands have two functional units: the secretory unit, called the Asini, and the conducting or excretory unit, called the ducts. The secretory unit. Cells of the Asini. The Asini are small sac-like structures. They are located at the beginning of the first segment of the conducting unit, called the intercalated ducts. The principal components of the Asini are mainly two types of cells: the mucous cells and the serous cells. The mucous cells. These cells are pyramidal in shape. A light staining, viscous secretory product called the mucus fills the cytoplasm of these cells, due to which the nucleus is flattened and appears displaced to the base of the cytoplasm. The serous cells. These cells are pyramidal in shape. They have round nuclei located basally. The rough endoplasmic reticulum is prominent in these cells, due to which the cells show basophilic stains towards the base. Secretory granules or zymogen granules can be seen accumulated at the upper end or the apical part of the cytoplasm. Hence, the apical portion of the cells take up a eosinophilic stain. These cells are responsible for secreting protein-rich watery secretions. Serous deminules. In some glands, both serous and mucous cells are present with the same secretory acini. Here, mucous cells predominate the serous cells and are seen assuming a crescent or moon-shaped cap over the mucous cells. These cells are called the serous deminules. The demilunes release their secretions into the lumen through intercellular spaces between the mucous cells. Myoepithelial cells. These cells are flattened cells that surround both serous and mucous acini and also seen in initial portion of the duct system. These cells are highly branched. They are contractile in nature and help to squeeze out the secretions into the lumen. The excretory unit. The intercalated ducts. The secretions initially drain into these ducts. These are the smallest ducts of the salivary glands. They show a tiny lumen with a lining of low cuboidal cells. The striated ducts. These ducts are formed as a result of merging of several intercalated ducts. These ducts have columnar cells which show basal striations. Excretory intralobular ducts. Striated ducts in turn coalesce to form excretory intralobular ducts. These ducts are surrounded by increasing layers of connective tissue fibers. Interlobular and interlobar ducts. The intralobular ducts join to form these larger interlobular and interlobar ducts. The parotid glands. The parotid glands are completely made up of the serous acini. Hence, they are also known as the serous glands. A section of parotid glands under the microscope show numerous connective tissue septae arising from the connective tissue capsule, dividing the glands into lobes and lobules. Within the connective tissue, there are nerves, lymphatics, ducts, and occasionally adipose tissue. Interlobular excretory ducts are also seen in the connective tissue. The serous cells in the acini are pyramidal in shape and are arranged around the lumen. Small secretory granules can be appreciated at the apical portion of the cells. The nuclei are round to oval and are located basally in slightly basophilic cytoplasm. All serous cells are surrounded by the myoepithelial cells, but owing to their small size, they are not always visible in some sections. The secretory cells empty their secretions into the intercalated ducts, which in turn empty the secretions into the striated ducts. From here, the secretions are emptied in the interlobular excretory ducts. The submandibular glands. The submandibular glands contain both serous and mucous acini. Hence, they are also called the mixed salivary glands. The serous acini in these glands are similar to that of the parotid gland, and the mucous acini are larger than the serous acini and show larger lumina. The cells are columnar with pale cytoplasm and a flattened nuclei pushed towards the base. Serous demilunes can be appreciated within the sections. The duct system and the connective tissue show similar features to that of the parotid glands. The sublingual glands. The sublingual salivary glands are predominantly made up of mucous acini. This marks a differentiating feature between the submandibular and sublingual gland. The ductal system and connective tissue show similar features to that of the submandibular gland. Difference between serous acini and mucous acini. Serous acini. Lumen is small. Mucous acini. Lumen is wide or large. Serous acini. Shape of the cell is pyramidal. Mucous acini. Shape of the cell is pyramidal or columnar. Serous acini. Nucleus is round and basally placed. Mucous acini. Nucleus is flattened, pushed towards the base. Serous acini. Staining is eosinophilic apically due to the presence of secretory or zymogen granules. Basophilic at the base due to prominent rough endoplasmic reticulum. Mucous acini. Staining is pale or lightly stained due to mucinogen granules.